that and even the Salk then kind of partially retracted and modified their statement, and they said, well, what we've claimed about direct cytotoxicity associated with spike applies to the native spike, but it doesn't necessarily apply to the vaccine spike. But they don't actually do any studies to show that it doesn't apply to the vaccine.
But they don't know.
Thank you. Okay.
I mean, that's all.
And I try to live in the world of, do we have data? Right? We shouldn't make assertions about whether something's safe or not safe unless we can demonstrate it.
Yeah, I mean, this is what you're supposed to do before you take it to market. You're supposed to...
Before you even put it into humans.
Yeah.
Okay? You're supposed to do this. And another thing that you're supposed to do is the belief system that just because we put a tag on the spike protein that's a membrane anchor, that we think it's going to make it anchored into cells, it's not good enough to just say, we think we did it, it looks like we did it, that's good enough. Okay? No. What you got to do is you got to prove the point. Which is why this Harvard and Brigham study of nurses that came out, in which they detected free spike after vaccination in a large number of subjects with very sensitive assay, suddenly rocked the world. And I mentioned that also in the Brett Weinstein podcast.
What are the implications?
That the spike was being expressed in cells, it was being cut off of those cells, and then it was circulating widely in the blood and presumably the rest of the body. Okay, so now you've got spike. And when I first got this data, I ran the numbers, and it only comes out to about 1/3 of a microgram total of spike protein that's detected as free protein in the circulation in these nurses at peak. Doesn't sound like much. For a highly biologically active protein, I mean, when I saw that, I thought, holy moly, this is the most successful non-viral gene transfer method I have ever heard of. I never thought that anything like this would happen, that they would get this level of expression. You know, to have that in your whole body, that's not a tiny amount of protein. And then when you think it through, it's just the tip of the iceberg, because you've got the spike that's still attached to the cells, and the spike has a ligand. It binds to ACE2, okay? And ACE2 is everywhere. It's in your vascular endothelial cells. It's all over the place.
This is why myocarditis, pericarditis, and these other...
That seems to be more of a coagulopathy, I think. That's another problem. Okay? But yeah, spike goes all over, and the amount that you detect as free protein is probably just a tiny fraction, because it's in equilibrium with bound to ACE2 protein, which is going to be a big sink, and that's in equilibrium with a spike that hasn't been cut off of cells yet. Okay? So when no one's ever measured all this stuff, which gets into another one of the huge bear traps here in terms of what the FDA did. If you could take the Pfizer dossier, common technical document, as revealed by Bridle from the Japanese government as face value for what they knew at the time when they moved this into humans in a big way. They didn't actually test the final drug product. They used luciferase to look at biodistribution. Remember I was talking about luciferase?
Last one passed, yeah.
It's the same basic construct, okay? And they didn't actually use the final formulation you're supposed to use. If you look in the guidance, everything I've always been taught and what I've always thought we had to do, you have to use a near GMP or GMP manufactured final product to do the pivotal toxicology tests of biodistribution, duration of expression, cell location, all this kind of stuff. They did none of that. The governments just let them get away with it appears, taken data off the shelf that they had developed for other purposes and kind of slammed it all together and bless it and off we go. As a consequence of that strategy and not insisting that the gene therapy checklist be applied, we have no real information about how much protein is being made, where it's being made, and for how long.
So we're flying blind in essence, and we're basically is the idea that we're going to just put it out in the world and then we're going to collect our data after we do it. I mean, it seems irresponsible.
I concur. That's why I had made the threat assessment that we should focus on repurposed drugs, because to do it right is going to take a long time. Yeah. And furthermore, to establish safety, when there's this history of antibody-dependent enhancement with vaccines in general, which often manifests over time, usually you need at least a year's data, usually 2 years' data, after you've administered to a very large number of patients, willingly accepting that participation in those clinical trials, not forced or enticed.
Yep.
And you have to follow them rigorously to make sure that they don't develop long-term adverse events like autoimmunity. They just flushed all that. And the other thing that they did that's kind of shocking to me is the FDA had the statutory latitude to require rigorous characterization of safety and efficacy during this period of emergency use authorization. Now, to my knowledge, they've only used EUA once before for a vaccine. That was for anthrax vaccine. for the Ebola vaccine, they used something called expanded use access. And with expanded use, it required that the pharmaceutical company, Merck, capture rigorously all data on adverse events and efficacy. And that is what, in large part, allowed the prosecution and licensure of the Merck vaccine. After years, the FDA gave them all kinds of hard time, but they did get through it because they had this safety database that was quite rigorous. In this case, the FDA basically gave the pharmaceutical companies a complete pass. And they said, we're not going to ask you to do anything in terms of safety follow-up. We're not going to require, even though they said in their emergency use authorization that antibody-dependent enhancement was a risk. They said it's a risk, it remains unresolved, and basically they said it would be nice if you would do some targeted studies to rule out antibody-dependent enhancement, to which, near as I can tell, the pharmaceutical industry basically said, well, thank you very much, we'll take that under advisement, and they did nothing.
What is an antibody-dependent enhancement?
So it's one of a spectrum of processes whereby a vaccine causes enhanced disease. It's one category. It's the one that's easiest to discuss.
Basically, it's like it's an amplifier almost. Saying you got the disease and now it actually amplifies it rather than.
So a key nuance here. Remember, going back, it's important to kind of parse this because So I've had a recent correspondence from Janet Woodcock about this, the current acting FDA commissioner. And she says, well, antibody-dependent enhancement is not happening because the disease doesn't seem to be worse in the people that have been previously vaccinated that get then infected the breakthrough infections. The disease, remember, is your body responding to the virus.
Right.
The hallmark of antibody-dependent enhancement in the context of this particular virus, when you think about it, is going to be increased levels of virus replication. That's the measurable thing.
That's the enhancement. Yeah.
Right, okay? It's not the disease enhancement, it's the virus replication. And what are we seeing with Delta? Oh, we're seeing levels of viral replication, even by Dr. Fauci's own mouth. But the studies are coming in even more now. The levels of virus being produced in the previously vaccinated with Delta, in the infected subjects, so the breakthrough infections, are at least as great as those that have not been vaccinated. And in some cases, there's evidence that they're higher.
So by definition, that's it.
This is it looks like a duck, quacks like a duck.
Get the duck.
Okay, at least from my standpoint.
Yeah.
Now, the numbers are small of samples. A lot of it's coming from Israel right now. The United States isn't even testing for Delta. They're just saying virus or no virus, assuming it's Delta.
So the reality is there's some evidence that maybe AD is happening, and there's really not enough research to say that it wouldn't happen, to disqualify it saying that's a concern, right?
So the truth is, in the FDA's own documents, and then in this recent correspondence that I saw from Janet Woodcock, the acting director, when the FDA granted emergency use authorization to Pfizer, in their summary document, they specifically said that antibody-dependent enhancement was a risk, a known risk, and that it could not be evaluated based on the data that they had provided. And they encouraged that such studies be performed, clinical studies, but they did not mandate those studies. I think that was another one of the major oversights, regulatory oversights, of basically giving a major vaccine manufacturer a pass. Why did they do that is speculation.
Did you talk to your friends at the FDA? And
Not about this one.
Okay.
Yeah.
I'd be curious because you obviously have worked with these people or crossed paths with them, at least as colleagues over time. And so, and I don't want to ascribe any speculation around conspiracies, this or that, but no doubt, let's talk about agenda. There was an agenda, an obvious agenda, an overt agenda to get these vaccines out, and somehow
Yeah, it was not subtle.
it was not subtle,
Right? It was stated government policy. There was a lot of messaging in the media that no shortcuts were taken, but it's self-evident that a process that normally takes a decade to do it in a matter of months, there will be shortcuts taken. What's rolled out over time is the depth and breadth of those shortcuts is profound. And for whatever reason, the willingness of the regulatory agencies, and it sure looks like regulatory capture. It's what you would expect in a regulatory capture environment where you had internal government advocates that were pushing for a particular outcome. It's very clear that standard norms that would be implemented for any other vaccine in any other context that I've ever known were overlooked. And they had to do with safety. And I haven't been into the data as deep as some people have in the clinical trials, but I hear again and again about oddities in those clinical trials and their interpretations. They were very abbreviated trials, and what I noted in looking at the trial design, as someone who designs trials for a living, is that after phase 1, they dropped the 14-day bleed, which is the discrimination for whether you're getting a primary immune response or a recall immune response. And all the evidence is that we're getting recall immune responses against prior coronavirus infection.
What does that mean?
So what it means is there's some deep stuff that it means. We've all had these, we call it the common cold, these circulating coronaviruses. And there's enough overlap in terms of their the immune response that's generated against those with SARS-CoV-2 that antibodies against those viruses and cellular immune response against those viruses are provoked when you get infected or vaccinated with the COVID vaccines or infected by SARS-CoV-2. Okay, so this is called a recall immune response. One of the practical consequences of this that really hasn't been adequately addressed right now, well, one of the early ones is we heard all of this talk about neutralizing antibodies. Neutralizing antibodies are not a correlate of protection. They haven't been proven to relate to anything relating to whether or not a vaccine will protect against you.
Right.
I don't know if you recall all of that buzz that was happening about a year ago now, where we were hearing that this neutralizing titer was higher with this vaccine versus that vaccine. It was like a, who's this bigger kind of thing was going on between the different manufacturers. It was all noise. Those neutralizing antibody responses were already known to be provoked as recall responses after infection.
Right.
And so you're amplifying the reactive cells, B and T cells, that were previously educated during the prior infection, and you're causing those to expand. Now, what are the problems with that? Now we go deep immunology, forgive me, it's called original antigenic sin. I love the phrase. I'm glad to get it on camera. But what it is that when your immune system is primed to respond in a certain way to a prior closely related infection, and it receives a signal from a new pathogen that's closely related, the immune response will be dominated by the reactive memory cells that were educated from the prior infection. And they will partially block the ability to develop new responses against the new pathogen, okay? And furthermore, they cross-react in these cell culture and in vitro assays that are really crude. And ELISA is an extremely crude test because it's a mixture of any antibody that cross-reacts with the antigen, whether it's functional or not, good, bad, indifferent, it all scores as positive.
Not specific.
Okay? And so it's really misleading. And then these neutralization assays are the ability to block either a pseudovirus or a live virus in cell culture with a defined cultured cell line that really is a long ways away from whether or not it has anything to do with your body. And in the real state in which you've got all of the things going on that are going on in your body. And the honest truth is that vaccinologists like to tell ourselves that we're so sophisticated and we've all got all these great assays. A lot of them developed during the AIDS years. And if we take a good hard look at ourselves in the mirror, the truth is that we're deceiving ourselves about a lot of that stuff. It's been one of the core problems is we've assumed that the assays that we've developed are measuring something that really matters. And that's part of what prompted this monoclonal antibody excursion that I talked about that did the mapping, was the discovery with Ebola that a lot of the antibodies that are neutralizing don't work for beans, okay? They don't protect. And other antibodies that are non-neutralizing turned out to work really great. So it turns out that we were fooling ourselves and throwing away the baby with the bathwater probably for years and years and years because we convinced ourselves that we had an assay that related to protection that didn't.
Well, that's
kind of what we had going on in the early days with this one.
Well, is it true now that also, so if I'm interpreting correctly, just because you have antibodies in the blood or so-called humoral immunity, that does not translate directly into cellular immunity, is that?
That's true.
Yeah.
Okay, and as we use the terminology, so this is the T effector cells versus B cell antibody-driven responses, and then there's the other part. This is right out the edge of modern immunology now, is we always assumed that innate immune responses, okay, innate immunity, this is our lizard brain version of the immunity, right, that goes back phylogenetically, with the one name that's associated with this that some people might recognize is Polly Matzinger and her danger signal hypothesis. Now we have what are called PAMPs and DAMPs, so pathogen-associated molecular patterns and danger-associated molecular patterns and blah, 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 that we have detectors for, but we also have natural killer cells. And it turns out, to my surprise and many others, that in fact the innate immune response is also adaptive. Okay? So when you get a vaccination or you get an infection, you're not just tweaking the B cell compartment, that's the antibody-driven group, or the T cell compartment, which, by the way, interacts with the B cell compartment, but is famously associated with cytotoxic T lymphocytes, the things that go around and hunt for cells that are infected by virus and kills them. Whereas the antibodies mostly are about binding and neutralizing antibodies, or there's some antibody-dependent cytotoxicity, but that's kind of more, that's like original antigenic sin, higher level immunology. So now this third arm that we thought was kind of passive, and it was just there, has got an adaptive component. Now, when we talk about the problems with universal vaccination, remember what I just said to you. Okay, we'll come back to that.
That completes part 2 of my 3-part interview with Dr. Robert Malone. Don't miss part 3. We cover some very significant and important ground there. You're going to want to see it. Thanks for being here. I really like people who are unabashed, people who just tell it like it is, not that they're trying to do it for effect, it's just their nature to be forthright. And when you're dealing with really smart people like neurosurgeons, it even gets better, and that's exactly who Dr. Jack Kruse is, a neurosurgeon who is unabashed, who certainly has very clear and well-reasoned perspectives when it comes to COVID. So I'm excited to share this interview with you. Dr. Kruse, thanks so much for taking the time to have this conversation with me. I'm looking forward to hearing your thoughts around this subject. Let's start with your background, as far as your academic background and kind of how you got into doing what you do today.
When it all started out, I was actually a dentist, got my degree at the University of Connecticut School of Dental Medicine, then went into oral surgery down at LSU.